This seed has a father, but genetically no mother. That sounds impossible until you follow what the word seed is actually hiding. The tree is the Tassili cypress, Cupressus depresiana, growing in the sandstone mountains of Algeria's Sahara. It is a living relict, a cypress surviving in a landscape far drier than the forests we normally associate with cypresses. Start with the usual picture. Pollen brings one nuclear genome. An egg brings another. Those two contributions combine to make the embryo, while the surrounding seed protects and feeds it. We casually call both contributors the parents because, most of the time, that model works. The Tassili cypress breaks that expectation. Its pollen can be unreduced. It carries a full chromosome set instead of the reduced set we normally expect from a sex cell. More unusually, that pollen can behave like an embryo starter. Scientists call this paternal apomixis, or androgenesis. It develops inside an ovule, but the maternal egg nucleus is not supplying the embryo's nuclear genes. Here is the detail that keeps this from becoming a magic trick. The ovule is still there. Its tissues form the seed's protective and nourishing environment. Think of the embryo and the nursery as two different jobs. In the documented mechanism, the pollen donor supplies the embryo's nuclear genome. The ovule supplies the place and resources in which that embryo can grow. Researchers made that separation visible in a controlled cross. They put Tassili cypress pollen onto ovules of the closely related Mediterranean cypress, Cupressus sempervirens. The other species supplied the ovule environment, but the resulting embryo carried the pollen donor's nuclear identity. The cypress was not making a hybrid in the ordinary sense. It was using a surrogate seed parent. This was not inferred from a strange-looking seedling. The researchers compared seedlings using morphology, chromosome-level ploidy measurements, and genetic markers. Those tests supported a pollen-derived, all-paternal embryo. In the reciprocal direction, the Tassili cypress's ovules could also support embryos from foreign pollen in the experimental setting. The exact cross is a laboratory result. It does not prove that one species routinely borrows the other's ovules in the wild. Now zoom out. A clever seed mechanism still has to produce enough new trees. The Tassili cypress survives as a tiny relict population scattered through hard-to-reach gorges. The last complete inventory cited by conservation sources counted 233 living trees across 46 sites between 1998 and 2001. That is an important historical baseline, not a current census. And recruitment can fail before a seed ever becomes a tree. A 2026 field study reported a cone-moth, Pseudococcix tessellitana, in the wild population. The researchers found the moth in a substantial fraction of the sampled cones, with infested cones containing far fewer apparently healthy seeds. The exact figure was 30.61% of the sampled cones. That number matters, but only inside its boundary. It describes the study's cone sample, not every tree in the Sahara, and it does not prove the moth caused the species' long-term decline. It does show a concrete place where a fragile recruitment chain can break, inside the cone, before the next generation is visible. So the Tassili cypress is not a tree with no mother in every sense. It is a tree whose embryo and seed nursery can have different biological roles. Pollen supplies the embryo's nuclear genome. Ovule tissue supplies the protected place where it develops. A controlled surrogate cross makes that separation visible, and the new cone-moth result reminds us that an elegant mechanism is not the same thing as a safe population. If you want to understand an extraordinary reproductive claim, ask two questions: which tissue supplies the embryo's genes, and which tissue supplies its nursery? In this Saharan cypress, those answers come apart, and that is the real biological surprise.